Ladies and gentlemen, it's a huge pleasure to welcome David Edwards. David has been the Weston professor in clinical sciences, mostly working on the Hammersmith campus, and I had the privilege of being a close working colleague with David for a long time. And it's difficult really to talk about him without embarrassing him or embarrassing oneself. I don't know of another clinician who is a greater role model as a clinical academic. David's extraordinary humanity is manifest by the amount of time as a busy researcher he spends looking at and trying to care for and deal with these very small babies, which are so precious to individuals in our society and to our society. It's a most difficult area of medicine, and as he said once in an interview, that it's a deeply distressing area. And he has shown, to my certain knowledge, an extraordinary compassion in that field, as well as immense leadership in a unit which is one that is absolutely a front-running unit anywhere in the world for dealing with very small babies. The work that has gone on at Hammersmith is probably as good as anywhere that I can think of. Is really a very, very important unit, and he has done a huge amount to lead that and to show ultimately that the things that really matter are the babies and their welfare, and the immensely difficult ethical considerations that arise from trying to decide whether you rescue a baby that might be brain damaged and its long-term future when it's perhaps only delivered at 24 weeks or 25 weeks gestation and very tiny. These are not easy problems, and they're ones that are still focused on. Some of you may have listened to Joan Bakewell this week in the ethics committee, and I thought about David's work when I heard her speak. A long introduction isn't appropriate with any speaker, but I do want to say that the work that David's done, which he's going to talk about, brain calling, brain damage, particularly in premature infants, is not only prize-winning, which it indeed has been, but also is a hugely significant advance. Because for my money, the reason why it's so important is because it's an example of where you can use very high technological investigation to develop a very low-tech possibility of treating people on a wide basis. And David combines the low-tech with the high-tech in no small way. And I think we all know that David is now moving on to another university south of the river. It's interesting being south of the river. We hope that it's not an entire loss to Imperial College. Whatever happens, it certainly won't be a loss to medicine, nor a loss to clinical science. I don't know of anybody, David, who's been a greater role model for me. And so, thank you very much.
It's a bit difficult to follow that, actually. As far as I can see, my job isn't any of those things. My job is to keep you out of the pubs for half an hour. Andrew thought, I think, that it was not appropriate that the graduate school should all end up in the bar before 5:30, so I'm here to stop you getting there. I think what I am going to try and do in that half an hour is to put work in context. I've had a happy afternoon browsing around the posters and trying to understand them. And it's very clear that there's a terrific amount of very high-quality science going on, and that all of you are putting your life and soul into producing a piece of knowledge, a really qualified piece of knowledge which is true and accurate and precise. And that's exactly what you should be doing. But I thought it might be helpful for a minute just to think about the context that that piece of knowledge goes into and how you turn that knowledge into something which is valuable. As Robert said, I deal with babies. Babies are supposed to look like this. They're supposed to be wrapped in lace and look fine. And this is a very deep human belief. If you go back to way back in history. This is the 15th century. This is a French illuminated manuscript written by a man, or painted by a man called the Boucicaut master. This is just a picture of a child being born. This child's been born, it's been wrapped up in swaddling clothes, and it's being handed to its happy mother, who is, as you can see, perfectly compos mentis. Everything is fine, the baby's wonderful, and everyone's happy. Even earlier, the 13th century, this is a picture of the Virgin and Child, which is one way our society characterizes the mother-child relationship, or has done. And you can see that even if you're the Queen of Heaven, you still look at your baby with adoring eyes. And your baby, who might be the living God, actually still cuddles you because he needs to. And this kind of relationship is very profound in our society, and indeed probably in all societies. The man who painted this, by the way, has got this rather elegant signature on the bottom. That's him. He's painted himself crawling around the Virgin's feet. This is the same sort of thing. Parent-child relationships don't always go smoothly, as you can see here. Although, in fact, what's happening there is actually the young God is actually forgiving his mother for the apple which he's holding in his hand. Whatever, this is a very deep thing in our society. It still is deep. Picasso paints it beautifully, showing the mother looking at her baby. And this happens in real life too. This is just an ordinary mother in India looking at her ordinary baby, both of which are very special. And yet, every year, 7 million babies die and never get to look in their mother's eyes like that, or be looked at by their mother like that. And this almost all happens around the time of birth. Of those 7 million, 2/3 die in the first month, of those 2/3 die in the first week, and of those 2/3 die in the first day. And this is a terrible cull of humanity which causes immense grief, sadness, and of course, biological waste. The major causes of these deaths are actually fairly simple. About 1/3 are caused by infection. About 1/3 are caused by prematurity, babies that are born too early that haven't had the full period in the womb. And about 1/3 are caused by birth asphyxia. And even when these things don't kill babies, they are potentially extremely damaging to them. And these three major causes, infection, prematurity, and asphyxia, if they don't kill babies, still have the potential to cause immense and severe brain damage. And what I'm going to talk about today is birth asphyxia. We're not going to talk about prematurity, but birth asphyxia, which is, across the world, one of the major causes of human disease and suffering. And we're going to talk about it in the context of high science, because if you think about it, I could give you two babies. One will be dead, and one will be alive, and you'd be quite hard pushed at a quick look, or even quite a hard look, actually, to work out what was going on in either of their brains. Very difficult to see inside the skull. You can do it, actually, you can shine light into the skull a little way, but you can't really see what's going on. So you need tools to work out what's happening inside the body. And one of the most powerful tools that we have for studying living people is magnetic resonance imaging, or magnetic resonance spectroscopy. And this was first discovered in 1953 by physicists, Purcell and Bloch, who were using it actually not to look at tissue, but to look at atoms and molecules. They got the Nobel Prize in 1953, and then in 2003, Lauterbur and Mansfield both showed independently that this technique could be used to take pictures, used to make images. And this actually is an example of the coil that Lauterbur drew for his first paper in Nature, for which he got the Nobel Prize. And it just shows you his vision of taking an image of the whole body using what had originally been a sort of chemical balance, effectively, used in a laboratory. A man of great insight. Mansfield also, great insight. Mansfield said that when he was told he got the Nobel Prize, he'd been expecting it for so many years he'd given up, and he told his wife to go away and leave him alone to keep on papering the spare bedroom. But he really had got it that time. This technology is quite big, it's quite heavy, it's quite expensive. And what you see here is a magnetic resonance imaging spectrometer being hoiked up through the sky, over the roof, and through a hole in the roof of the Hammersmith Hospital. Our intensive care unit for babies is on the 4th floor, we're on the top floor. So we cut a large hole in the roof, we got a big crane, and we just hoik this thing up in a force 5 wind. You can see the rope swaying a little bit, actually. And the man who was running the crane, who was a remarkable man, he sat there in the cab, he had a flat cap on, a flag hanging out of his mouth, a cup of coffee on the dashboard, and 2 levers, and an earpiece. And a man on the roof was going up a bit, down a bit, right a bit, left a bit. I mean, that's all the information he had. And with that information, he put that 3-ton machine down within 2 inches of where it had to be first time. You had to admire his skill. The brain's a remarkable thing. That scanner is fully operational, and it's one of the type that's been used to unravel this problem of birth asphyxia. With a scanner like that, you get pictures like this. This is a baby who's normal on the left-hand side, with a little arrow pointing to a particularly useful sign of the internal capsule. Don't worry about what it is. And on the right-hand side is a very abnormal brain of a baby who's been severely asphyxiated. And I think even without being a fellow of the Royal College of Radiologists, you could probably see 2 large white lumps on either side of the brain. They shouldn't be there. That baby has a dreadful future ahead of him, and he may well die of that disease. This is a very useful thing, though. We can actually now look inside the head, look inside the brain, and begin to get some idea of what's really happening. And in fact, if you do that, you can show something very useful, which is that children who have this problem, who turn up looking as though they've had birth asphyxia, actually all have had insults which have happened very recently. For a long time, there was a great deal of worry that these children who appeared very sick when they were born with asphyxia had actually had things going on for weeks and months, and it was just turning up at birth. It's not true. These images show quite clearly that these lesions were acute. They'd happened very recently, and if they're acute, we were close to the origin of the disease. We might be able to do something about it. And this is just a paper that came from our unit. Francis Cam was the lead author, which showed this about 10 years ago. So, a little biochemistry. The other great thing about magnetic resonance is it doesn't just take pictures, it allows you to look at biochemistry. And this is the level of biochemistry which I understand. As I told Robert earlier, I don't actually read Nature and Science or difficult journals. I just do the easy stuff with the babies. But I do understand a bit of biochemistry, and I understand that ATP is the energy currency of the cell. And that with ATP, you can do anything you want to do. And without it, you can't do anything at all. You can't maintain your membrane potential, you can't think, you can't digest, you can't do anything unless your cells have ATP. You can't move. And ATP is normally made by oxidative phosphorylation and glycolysis, and that's a very effective system which works very well. Highly efficient, very good at turning food into energy. But there is a problem, which is your cell only has a few seconds worth of ATP in it. Now, if you block oxidative phosphorylation and glycolysis, and you're being chased by a lion, your muscles will run out of ATP after a few steps. Now, that's not a very evolutionarily stable situation, and it's difficult to believe that the human race would have survived if its ATP runs out as soon as it's chased by a lion. The lion will be the same, of course, but we'll not worry about that. And in fact, nature isn't stupid. Nature has a backup system. Nature doesn't provide much ATP in the cell, but it does provide an energy source which you can use to make ATP without oxidative phosphorylation, which is phosphocreatine. You can break down phosphocreatine inside muscle or brain cells and produce creatine and make ATP again instead of regenerating your ADP. If you do that, then 2 things happen. The first thing is that your level of phosphocreatine falls. The second is your level of inorganic phosphate, which is a byproduct, rises. Now, magnetic resonance spectroscopy has the great advantage that it can measure both of those species. It can measure phosphocreatine and inorganic phosphate. And therefore, if you can measure inside a brain the fall in phosphocreatine and a rise in inorganic phosphate, you can be pretty certain that the oxidative phosphorylation pathway is wrecked. It's not working. And that turned out to be the key that unlocked brain injury for newborn babies. And I show there a spectrum showing 3 ATP peaks and a normal phosphocreatine peak from a baby acquired many years ago. But you can see that you can see very easily, with a very large peak, how much phosphocreatine there should be in the brain. Osmond Reynolds at University College Hospital in the 1980s realized this, and so he acquired an MR spectrometer and started studying babies with it. And he acquired this data here, which, as you can see, because this is an old Blue Dazo slide, is very old. But it is the key slide which has unlocked the problem of brain injury. On the x-axis of this slide is the age of the baby in days after birth, and on the y-axis is the phosphocreatine and inorganic phosphate ratio. So the higher that is, the more normal oxidative phosphorylation is in the cell, and the lower it is, the more damaged it is. The CL line is the lower confidence limit, and the M line is the mean line for this species at this age. And what you can see is that the phosphocreatine inorganic phosphate, which I shall call PCR-PI in future because it's much easier to say, the PCR-PI ratio falls in these babies. Now, these babies were all asphyxiated. They were all children who'd been undergoing serious problems in the womb. They'd been often untimely ripped from their mother's womb by well-meaning obstetricians through cesarean sections. They'd been delivered in a terrible state. The pediatrics team had been there. They looked extremely sick. They'd gone to intensive care. And then as soon as possible, they'd been taken to the scanner and looked at. So they're all very sick babies. And there's one remarkable thing about this slide, which you may have spotted, which is that the first measurement is normal. The first measurement at about half a day is normal. It hasn't fallen. It's within the normal confidence limit. And then it falls. And that single insight, that single dot on this graph, enabled people to think about brain injury in a new way. Everybody up till this time had thought that brain injury occurred as soon as you were starved of oxygen. If you're starved of oxygen, your cells die, that's it, nothing you can do about it. It's not true. That's not what happens. It seems that there's a time before the brain cells start to die, a time before the energy processes are wrecked. And that's shown on this graph very graphically. This lower level, though, this fall, if it's meaningful, if this is really what matters in the brain, this really should tell you what happens in the future. So this, you can make a prediction now, which is that this level, this value here, should predict the child's outcome. If it's very low, this child should be badly damaged. If it's not very low, the child should not be badly damaged. So you can form a hypothesis and you can test it. And this is the result of that test. These are children who were followed up till they were 2 years and then 4 years of age. And to cut a long story short, if these children had normal PCR-PI ratios when they were born, then they were normal when they were 4. If it was very low indeed, they died. And if it was middling low, they had severe cerebral palsy. So this low level, the level to which this PCR-PI value falls, the level to which your inorganic phosphate ratio demonstrates energy crisis, does predict the future. It predicts what's going to happen to you when you grow up. It is important in how your brain works. If you look more into this delayed phase of damage, if you like, then other things can be seen. The first one is that lactate goes up. This is another measurement you can make with MRS, another one of Persson Bloch's discoveries, which is you can measure lactate in the brain. The top slide here, top spectrum, is a child that's been severely damaged, and the ATP's all been wiped out. And the lower slide shows a normal child shows the same child measuring lactate. There's a very high level of lactate in that brain. And the higher the lactate is, the worse the children do. And in fact, it goes up in exactly the same pattern. Over the few days after delivery, lactate in the brain slowly increases. This actually is a piglet, but it's the same sort of thing. It also predicts the future. So we have a hypothesis now about how brain damage in babies happens. We believe that they go along quite happily in utero, that before they're born, there's a problem, something goes wrong, the placenta comes away, they get some terrible damage that stops oxygen getting to the brain. Then they're delivered, and then we can start to measure things. And this is what we have here. So we have a period of asphyxia before they're born, then a latent period when things are ostensibly normal, but bad things are going to happen, and then a period of delayed cell death. And that hypothesis is the basis of all the work that we've done. If you have a hypothesis, you have to test it. So these are 2 tests done in animal studies of that hypothesis. One is with little rats, baby rats, who were given a focal stroke. And the other one is with baby piglets, who were given a global brain injury, in both cases by reducing the amount of oxygen and blood getting to the brain. And without making too big a point about it, if you look at this line here, then this is the normal control piglets, and they just go along quite happily with nothing changing. The PCR-PR ratio doesn't change at all. This control piglet here, this intervention group, they had their carotid arteries tied at this point here. I've lost my arrow. Oh, here it is. The PCR-PI fell dramatically because there's no oxygen in the brain. There has to be an energy crisis. Then they were given oxygen back again to simulate delivery. Everything went back to normal, and then slowly, over the next few hours and days, the brain starts to get damaged. And the same happens with a focal stroke in rats. You can see that a bit more graphically on this slide here, which is an image of the same piglets. This is piglet brain. I appreciate it doesn't look like a piglet brain. It's the top of the piglet brain. The rest of the brain's down here somewhere. And there's two little ears poking out the side if you had the whole picture. This is the cingulate sulcus of a piglet. These are diffusion images, and they show acute membrane failure as we go through the slide. This is the control slide baseline here. Asphyxia occurred at this point, and then the piglet was resuscitated. And 3 hours after resuscitation, the brain looks normal. 7 hours after resuscitation, it looks almost normal. By 10 hours, there's a little black snake crawling across the surface of the brain, which by 14 to 17 hours has begun to overwhelm the brain tissue. And by 45 hours, the whole brain is black. In this image, black brain is dead brain. So you can see that even that using imaging or measuring chemicals, there's a delay before brain damage kills brain cells. That gap is the therapeutic interval where we can do something. Once we realized this, there was a race to work out what to do. And a race to work out why. And the why is very simple, actually. The why is really simple, that unlike me, and probably even you, if I have a stroke now, most of my cells will die by necrosis, and that's it. Babies are different. Babies have a rather different cerebral biology. They're tuned on to apoptosis. As you know, nature isn't a potter. Oh, Mikhaim got it wrong. Nature doesn't take a lump of clay and mold it to the right shape. It makes a block of cells and then chips away like a sculptor. And that chipping away, that sculpting, is actually by apoptosis, by programmed cell death. Cells that are not needed die. So the newborn body, and in particular the newborn brain, is tuned to lose cells. In your brain, you lose about half the brain cells you make around the time of development. So the apoptotic pathways are turned on in newborn brain cells, and asphyxia seems to trigger them. It doesn't just seem to trigger them, it does trigger them. And the cells that are dying late, these day or two after an insult, are dying by apoptosis. Apoptosis is a biochemical pathway. Because it's biochemical, you should be able to interrupt it. How do you interrupt it? Well, actually, it's depressingly simple. You cool the cell down. If you cool the cell down, you will have a dramatic effect on its ability to apoptose, and you'll stave off apoptosis for quite a long time. And if you cool a baby down, you'll have the same effect. This is an animal study in the same piglets, just to show you the science behind it. This is the same kind of graph I showed you earlier. This is the energy failure on the y-axis, time on the x-axis. There's three groups now. The control group, which are the triangles, the insulted group, which are the circles, and then an insulted group, which then will cool down over this period here after the insult. Not during the hypoxia, but after the hypoxia. So we're trying to simulate now what happens when a baby's born. The baby's born, comes to intensive care, we start a treatment. This piglet was insulted, we resuscitated it, started the treatment. And cooling for 12 hours over this period here completely obliterated this secondary decline in energy metabolism. There are many, many studies that showed that. This is another one which actually looks at percentage brain damage. This actually is rats. Same story. Control animals have a lot of brain damage. 60% of their brains wrecked by this insult. Cooled animals, it's about 30%. You can halve the brain damage in animals. Okay, so to cut a long story short now, you're probably quite glad for that, aren't you? To cut a long story short, this is the result of 3 large clinical trials carried out to see whether this treatment would work in babies. We did 2 of them. We did the Toby trial and the CoolCap trial. The Americans, who are great, very helpful people, and when a good idea comes along, are very willing to replicate it for you, preferably before you do it. They did the NICHD study. These are forest plots. You're probably mostly familiar with forest plots, but some of you may not be. What they do is they display in a fairly easy to, or fairly inclusive way, the data from clinical trials in a series of clinical trials, and then produce a summary measure at the end of it. So the 3 clinical trials we're interested in are Toby, NICHD, and CoolCap. These are the number of patients in various groups here. The number of children, the total number there and there who had hypothermia or normothermia, and the number of events, which in this case is a bad outcome, and there and there. And then they're added up at the bottom. So you can see that there's about 700 patients in these 3 trials. The risk ratio over here tells you whether the treatment group was better or worse. If they're on this side of the line, then hypothermia did them some good. It favours hypothermia. If they're on this side of the line, then non-treatment was better for them. And you can see that in all three trials, the treated group did better. And if you add them all up together, you have a statistically significant benefit of treatment. And this is the data which shows that you should cool babies who've been asphyxiated. Cooling babies works. It reduces their brain damage and improves their outcomes. In purely practical terms, the numbers needed to treat to prevent death or disability is 9. And the inverse of that, or the not quite inverse of that, which is to create one baby who survives intact, is 8. Now, that doesn't sound very good. What it means is you've got to treat 8 people before one of them benefits from your treatment. But I'd say 2 things about that. The first one is that not all these children do badly anyway. We're not perfect in our ability to diagnose them. So in this group, about 5 of them would normally have bad outcome, and 3 of them would have a good outcome in the group we treated. So diagnosis isn't perfect. The second thing is that if you compare this to other treatments, it's not bad. When I was thinking about what the 10 great discoveries might be in the list that was going to come up, I did wonder if somebody was going to say statins, because statins are the great medical miracle of the last decade, really. They seem to treat everything and reduce death from a great many causes. And I know I should be taking it. I don't because I'm lazy, but I should be. And I'm sure many of the people my age here are actually taking statins. It does increase longevity and reduce cardiovascular events. The numbers of patients you need to treat with statins to produce one beneficial outcome is 100. And yet this is the great public health advance of our decade. So this is better than statins. So that's not bad. However, it's not perfect. And you still have a large number of children who don't benefit from it and either die or survive with disability. And so we aren't there yet. It is cost effective, mind you. I show you this journal because you've probably never seen it and never will see it. It's called Value in Health. It's an health economics journal. It's absolutely incomprehensible. If you think the science paper was bad, this is worse. And we wrote it, and I don't understand it. This paper is a proper economic evaluation of cooling. I sort of understand it after 4 or 5 rewrites, but it does show that it's cost effective. This has saved the country about £100 million so far. And the numbers of babies being cooled is going up dramatically. We run a register for babies who are cooled. This is now quite a long way out of date. We've now cooled about 2,000 babies, and you can see the number of sites doing it, the number of patients being cooled is going up. I want to stop now and just reflect for a minute how we've got to this position, because when you're thinking about your own work and you think about the detailed experiments you're doing, the detailed studies you're doing, you may not appreciate just how dependent you will become on other people to implement that work, or how long it might be before it actually does any good. The first paper that I referred to today about delayed cell death came out in The Lancet in 1984. The first paper which showed any benefit from cooling post-ischemia came out in 1989, and I would say we didn't do that. I'm not claiming credit for that. That was done by Marion Ginsberg's group. It was done in adult animals, and he showed that if you cooled these adult animals after they'd had a brain insult, they did better. I think he was surprised by this. I think he'd actually set this group up as a control group to another treatment he was doing. And I think he was very surprised to discover this actually worked. But being a good scientist, he published his results. And, of course, those of us who are interested in trying to find a treatment jumped on it immediately. So credit for the discovery should go to Marion Ginsberg. The paper's Busto et al., 1989. From then on, my colleagues and I and our friends around the world got to work. We did a lot of animal work, some of which I've shown you. We did a lot of disease characterization and tried to work out how to run a clinical trial, because that wasn't immediately obvious. We did some pilot clinical studies. We've now got to about 2,000. Then we ran some big studies, the CoolCap study, the Toby study. When we'd done that, we did a meta-analysis, which I've just shown you the results of. Then we set up a registry, did the cost-benefit analysis, and by now we've got to 2009. And in 2009, NICE, the National Institute of Clinical Excellence, approved hypothermia for use in the NHS, and it's now used across the NHS and indeed across the world. That's 20 years. It's 20 years of a whole series of different scientific skills. Experts in magnetic resonance, experts in animal studies, experts in apoptosis, experts in clinical studies, experts in statistics, experts in economics. It goes on and on and on. You need all these experts to get these things to work together, and we all have our little place to play in these things. What do we do now? Well, regrettably, we go back to the beginning, because this treatment doesn't work for everybody. There are still children suffering out there who've not been effectively treated, and they weigh heavily on our conscience, and not they weigh heavily on their families. What do we do now? We're going to do it again. There's a whole series of treatments, because what cooling is more than anything else is proof of concept that treatment is possible. Nobody believed this was possible 20 years ago. I remember having a long discussion with a very eminent neurologist in our unit who told me I was wasting my time and we shouldn't bother. I love this story. Wasting my time and I shouldn't bother. It was complete terrible. I should do something completely different and I should go into genetics. I reminded him of this not long ago. We're going to go back to the beginning again. We've got more treatments. We've got Xenon I'm going to come back to in a minute because it's an Imperial College discovery. But melatonin is an interesting compound which seems to be very neuroprotective. And TRP601 is an interesting caspase 2 inhibitor that our laboratories just characterized that we think might be useful too. But we're not going to do it the way we did it before. We're not just going to run through that long program of prototype discovery, early clinical trials, late clinical trials, the way we did before. We're going to do it in a more clever way. We're going to use biomarkers to select out the ones that work. We were very lucky with hypothermia. We weren't that lucky. It was fairly obvious which to do it, really. But if we'd chosen it wrong, we'd have spent 20 years and wasted our time and a lot of money and a lot of babies' input. In the future, we're going to do it more cleverly. We're going to do it with biomarkers as an early phase in small numbers. It took 700 patients to prove hypothermia worked and many years of work. If we can do it in 20 patients and show a drug works, then we've got really good evidence to go and do the big clinical studies. If it doesn't work, we'll throw it away and try something else. But we're going to do it with biomarkers. What are biomarkers? Exactly what they say they are. They're things that measure a biological change. Examples are used every day in clinical medicine: blood pressure, blood glucose. There's clever things around like genomics, proteomics, metabolomics, and physiomics, some of which of you working because I've seen the posters. But actually, some of the most qualified biomarkers are imaging biomarkers, and that's what we're going to use. Imaging is extremely powerful. This is a brain atlas, which came from Imperial College. It's an example of how modern imaging can do things you can't even think of. This is Daniel Ruckert, who does some of this work. He's a computer scientist, and he's produced a core technology which enables all sorts of things to happen, like that brain atlas. He's invented the ability to add images together. Now, that may not sound very clever when you first think about it, but actually it is. Images are not numbers. You can't just go image 1, image 2 equals image 3. It doesn't work like that. They're not commutative. They're not associative. They don't have the property of integers. You can't add them. Daniel's worked out a way of doing that. And he's worked out a way of warping one image so it fits another. This is his equation. It's a bit more complicated than that in real life. This is a global transformation followed by a free-form local transformation. And this adds images together. Basically, what he does, he uses the mathematics of geometry, algebraic geometry, to move images around until they overlay each other. So if he wanted to do my hands, he would do one rotation like that, then a transformation like that, another rotation like that, and he'd pretty well have aligned my hands. The trouble is, these two fingers here aren't quite there. So if he now does that again, he can put those fingers right, but the other fingers become wrong. So my global transformations, which did that, need to be supplemented by a local transformation, which does that. And when he's done that, he's got the images completely aligned. At this point, and this is the clever bit, you throw the images away, because they're all the same. But what you've got the transformations that make them the same, and you work with the transformations. So you work with the mathematics. I'll show you this in practice. This is one of Daniel's films, which aligns this image to this one. Just watch this image go. This is an iterative process. By running his equations on that image, we've now made it the same as that one, and we can throw the images away and work on the transformations. This way of adding images together is immensely powerful. You can do things like this. This is a movie of brain growth. This is a whole series of babies' brains which have been added together, one after the other, to show them growing. And I like these movies because I'm interested in how the brain grows. You may not be, so we'll move on. This is how babies think. Well, it isn't really, but I like to put it like that. This is actually a really complicated slide, and I'm going to spend a moment on it because I think it's cutting edge. Actually, I won't spend very long on it. It's in Procnat Sci last year if you want to see it. Doria et al. This background grey image is a four-dimensional brain atlas. It grows as over time. Starts about 29 weeks and grows. It doesn't change size because it's been normalized for size. If you look at the brain structure here, you can see the cortex changing shape. Very simple cortex at 29 weeks, getting more and more complicated as time goes on. This is using Daniel's registration techniques to add images together. The red image that's on it is brain activity. This is the spontaneous brain activity that babies have at this age. And we've used the brain atlas to display the brain activity. And this is motor activity from the motor region. And you can see that over here, if you start over here, at 29 weeks, brain activity in the motor region is only on one side. As time goes on, it starts to talk to the other side of the brain. And this is just one example of how modern imaging can provide you with biological records of what's happening inside babies' heads. And I won't spend any more time on this. If you want to see it, it's in Doria et al. Procnat Sci last year. One really useful thing that this technique does, which we do use, and we're going to use for further studies of brain protection, is this, tract-based spatial statistics, which was invented in Oxford, not by us. And this is a really neat idea. What happens is, you take out the very center of the white matter in the brain, just one thin sheet through the white matter of all the images you're interested in. And then you add them together using Daniel's technology. And when you've done that, you've got an average image which has all the white matter in, completely uncontaminated by any grey matter around it. This is a very pure sample. It's like purifying your sample. It's like running your... do you run things on columns these days? I don't know. Anyway, it's like running your brain image on a column and getting out the bits you want at the bottom. You've just got the white matter. And that enables you to do statistical tests on it, which are very powerful and very precise. This is an example of that. These are two images, one of which shows the optic radiation going from the lateral radiculate nucleus to the optic cortex, and done by tractography, which is another imaging technique I'm not going to talk about. And then on the other side, the TBSS method, tract-based spatial statistics method, which has pulled out and purified the center of the white matter from those optic radiations. And to cut a long story short, where the background is green, I think it's green, I'm viciously colorblind. Is it green or red? It's green, thank you. The green background is just the white matter being shown. And where it's blue, there is a statistical relationship in that area of brain between the child's ability to see and the quality of the white matter. So the blue is showing you where there are problems in babies who can't see. This is a powerful technology for use in studying new treatments. In the randomized controlled trials I told you about, which proved that cooling works, we needed to study 767 patients to prove that cooling was effective. That's the gold standard end of study outcome. We know that it works from that. If, on the other hand, we used imaging and we took images of all these babies' brains and handed them to Mary Rutherford, who's my colleague, who's a very skilled radiologist, and asked her to tell us which ones were normal and which ones weren't, she could pick out the effect of cooling in about 130 patients, 131 actually. So that's a much better way of doing it than just waiting for the children to grow up. If you use this TBSS method, you can do it in 20 patients. That's a log order reduction in the number of patients and more than that in the cost of the study. So this is the method for testing new treatments which is going to be efficient and effective, which we're using at the moment. And I'll just tell you about Xenon quickly before I finish, because it's a discovery from here. Xenon, as you all know, is a no-ball gas. It has no chemical reactions at all. It has a completely filled outer electron shell and is inert. It comes as some surprise then to know that it's a profoundly biologically active atom. It has immensely strong effects on the brain. It's a very powerful anesthetic. It puts you to sleep. And Nick Franks, who was investigating this some years ago, found to his surprise that it blocks a channel, the NMDA channel in the brain, which is one of the ways that hypoxic ischemia kills brain cells. Hypoxic ischemia activates this channel, and because of that, it turns on apoptosis and cells die. This was a really important discovery, I think, and it gives us another opportunity to think about treatment. Because the great thing about Xenon is that it gets into the brain. It gets there very quickly, as the previous slide showed. Nick just got his FRS for this. If you give xenon to animals who you've given hypoxic-ischemia to and whose brains you've damaged, it's immensely neuroprotective. This is just one study. I'm not going to show you any more. There are a lot more studies than this. This is just one brain which has been sliced, and you can see the control brain is nice and symmetrical, nice and clean, nice and normal. If you reduce the oxygen that animal had been breathing to 8%, you get significant damage in the brain. You can see the brain's crumpled around here. It isn't not right. It isn't right on either side, actually. It doesn't right here either, and it's shrunken through. If you use 8% oxygen but give xenon as well, you get a remarkable effect of protection. And the great thing about xenon, apart from getting to the brain, is that it's synergistic with cooling. If you give the two together, you get more than additive effect, which is a real benefit. So we are at the moment running a trial giving babies xenon to breathe. It's an interesting problem how you explain to mothers you'd like their baby to be ventilated with an inert gas. But that's another part of the research program which I shan't talk about today. This is MRC funded. We're using biomarker outcomes. As with all research, we need some kind of commercial input. We can't do it just by ourselves as academics. Recruitment's going on at several hospitals, and we think we can do this in 60 patients, not the 700 we needed for cooling. So I need to acknowledge a lot of people who did all this work. Osmond Reynolds and the UCL group, I think, were the progenitors of all this work. Auckland were very quick on this, as were Oslo, and we ran trials with friends in America, New Zealand, all sorts of places. And the people at Imperial who've done this work, Dennis Azzopardi must come top of the tree. He was the research fellow with Osmond Reynolds when that initial work was done, and it's really largely to his credit that work came out. Joe Haynor and Daniel Rookert are physicists and computer scientists without whom we couldn't function. Mary Rutherford, Francis Cowan, clinical neonatologists and radiologists, and various other people who I can't go on to mention. But there's a big team of people here. This is the tip of the iceberg. There's about 100 people been involved in the work that I've been talking about now. I know that. I've just counted them. And that's leaving out a lot of the research fellows who deserve more mention than they get. I'd like to end just by pointing out to you that Galileo got it right. The laws of nature are written in the language of mathematics. Thank you very much indeed.